Welcome to everyone. It's a pleasure to have Professor Aditya Rumamurthy today as our seminar speaker. Aditya is a bright young researcher working in the areas of networking, communications, information theory. I've known him for a long time. I've known him for almost 10 years now. Just to give a quick introduction, he got his BTech degree from IIT Delhi in 1999, and then he got his master's and PhD degrees from UCLA. In 2002 and 2005. He worked for a little while at Biomorphic VLSI, and he also worked at Marvell Semiconductor for a while in the area of data storage. His interests are in the area of network information theory, channel coding, and signal processing for nanotechnology and bioinformatics. Aditya has been serving as the associate editor for the IEEE transactions and communications in the area of coding theory, right? Since November 2011. He received the 2012 Iowa State University's Early Career Engineering Faculty Research Award. He also received the Career Award from NSF in 2012. He also has a professorship, Arpal Palmer Professorship at ISU, I think in 2009 and 2010. Well, we're happy to have you.
Thank you. Okay, thanks for attending my talk. This talk is about maximum likelihood sequence detection, and I'll talk about two specific applications that we've recently been working on: DNA sequencing and nano-imaging. This talk is joint work with a number of people. So the Shen Yin, Zhao Song, and Karan Dorman, they are the collaborators on the DNA sequencing work, and Naveen Kumar, Pranav Agarwal. Naveen Kumar was my former student, and Pranav Agarwal and Bhuti Salapaka were at the University of Minnesota. And I'll talk about different places where what part of the work was with. So broadly, the idea of sequence detection, as we all know, is a fairly powerful idea, and it has led to numerous. I mean, it is a part of many major technologies like cell phones involve bit detection, convolutional codes. We have speech recognition engines that use a variant of maximum likelihood sequence detection in some form or the other, and also hard drives use sequence detection extensively. So it's a technology that has revolutionized a number of fields. Broadly speaking, the basic idea of sequence detection is that you are seeing a sequence of, you are observing a sequence of data which are noted by the Y1, Y2, so on and so forth, which are formed as a result of some sequence X1, X2, so on and so forth, that is being distorted in some manner. So the problem in sequence detection is that you're observing the sequence of Y's and you actually want to decode the sequence of X's, and there's roughly two parts to this problem. So the first part is to model the sequence X and the stated distortion in a probabilistic manner, and the second problem is to find the X that jointly maximizes the probability of observing X and Y. Okay, or so either you try to maximize this quantity if you can in a tractable manner, or some approximation to it. Now, I would like to say that ML sequence detection means different things to different people. So in some fields, we say that we are trying to maximize the conditional likelihood of observing Y given X. But here, for this talk, I'm going to say that it's joint likelihood, okay? Joint probability of X and Y. And depending on the application domain, both these tasks can be challenging. Okay. So what are some prototypical examples? The first is, as we well know, is one of inter-symbol interference channels. If you want to decode a bit stream that is formed by that is transmitted over a inter-symbol interference channel. So in this case, it's well known if you know the channel response and you know that the noise statistics are Gaussian, then the optimal detector is a match filter followed by an ML sequence detector. And this is due to classical results by Forney. So in this case, one thing is that we know the channel exactly, and we also assume some known structure on the input signal. Okay. Another prototypical example is in speech modeling and recognition, where typically you try to set up hidden. You try to model the speech using hidden Markov models corresponding to different words. You train different hidden Markov models corresponding to different words, and using this, then you perform not exactly sequence detection, but the forward-backward algorithm for determining which word was spoken by a given user. So in this particular case, you require some ground truth knowledge in training, and this training typically works with noiseless speech, and the recognition is again done by variant dependent sequence detection. So these are two areas where it has had a very profound impact. So what is this talk about? So this talk has two parts. The first part is going to be about error correction for DNA sequencing, and DNA sequencing, you probably heard the term, is the process of finding the identity and order of nucleotides in a DNA molecule. Okay. So let me say a little bit more about the first part of this talk. So this technology has been in the news lately because really the cost of sequencing has fallen dramatically. So here's a chart that I pulled off the internet. This is the curve in white is showing the Moore's law, the rate at which chips are becoming less expensive versus this is the cost of sequencing. And they say basically very soon you should be able to sequence your own genome for about $1,000. And this has happened due to this new technology called next-gen sequencing, which is driving a lot of this decrease in cost. And it has a wide range of applications, ranging from like personalized medicine to sequencing can also help you understand bacterial and viral action and things of that nature. And so the low cost really helps all of this. So the problem in DNA sequencing is as follows. So the actual genome sequence, for example, the human genome sequence is a very long string running into millions or billions of bases or nucleotides. But the DNA sequencer cannot actually sequence the whole genome all at once. So you need to infer the whole genome from short noisy reads, and you cannot really assume any data structure, and you have access to only noisy data because as you'll see, these reads are actually noisy, and there is lack of any data alignment. And I'll talk about that a little bit more. So in this picture, as you can see, you are seeing these reads that are coming as a result of reading some portions of the actual genome, but there's no alignment between them. Okay. So in this case, we'll show that sequence detection allows us to perform error correction and in fact clean up the reads. And I'll present results which show that our algorithms, something that we've been working on for about 2 years now, outperform all competing methods in the bioinformatics domain. And this is all with real sequencing data, okay, so real data. The second part of this talk is about how to apply sequence detection to nano-imaging. So here I'm going to talk about this instrument called the atomic force microscope, which is used in imaging at the atomic scale. So this is a schematic of the AFM. This is it has a cantilever that has radius of about a few nanometers, and it responds to atomic level forces. And so you do the imaging by bringing this cantilever close to your sample and interpreting the deflection signal appropriately. And it also has had a tremendous impact in the basic sciences. So for instance, it has been used to image the bond structure of molecules, chemical structure of molecules. IBM Millipede in the early 2000s created a data storage system using an array of AFM cantilevers. It can be used to image stuff at the image like real-time video at the atomic scale, and also it can also be used to create nano patterns, something that is not a part of this talk, but it can also be used to do something like that. And so in this talk, the second part, I'm going to talk about dynamic mode AFM, the certain mode in which this cantilever is used, which was traditionally viewed as a very slow method, okay, by physicists. And the main message of this talk is that in fact if you use sequence detection, we can actually perform AFM, dynamic mode AFM, making it a much faster speed than what was conventionally thought. So with that, I'm going to start the first part of this talk, which is about error correction for DNA sequences. So here's first I want to make sure the problem is explained very clearly. So here is the string that I have in the middle of the slide. You can just treat that to be the true genome. Of course, the actual genome can be much bigger. It could be of millions or billions of bases. And whenever I say base or nucleotide, it's going to mean the same thing for this talk, okay? And as I said earlier, you don't get to sequence this entire genome all at once. The reads only cover portions of the genome. So for instance, if this is the genome, then the thing that is in multicolor, this is like a read. Okay, so this read could be from a certain portion of the genome. Okay, so here I've shown you the correspondence. So this could be an instance of the first read. This could be an instance of the second read, and so on and so forth. So what I've shown here is a set of 6 different reads that are formed by reading different parts of this genome. Now, what's the issue that happens in the sequencing process is one of errors. So for instance, if you look at the third read here and the third base, so this base actually should have been called as a G.
Question?
Yes.
No, which is right, which is left?
I'm sorry?
Do you know which direction you are facing? As in your reads, is there directionality given or?
This is directionality. I mean, you know which. So this is the left side of the read and this is the right side of the read. Yes, you know that directionality.
You know that. Okay.
Yeah, and in fact that matters because in fact what happens is the reads are more noisy towards the right end of the read. So there's a 5' and 3' direction that both they're called. So yeah. So the problem is that this third read, the third base on this is miscalled. Instead of calling it a G, it's called a C. Okay, and the objective of error correction is to be able to look at your reads and actually correct them. So in fact, in this case, I would call this a success if I was able to correct this particular base back to a G. Okay, but I want to emphasize that it's different from sequence assembly. In sequence assembly, you are given these reads and you're trying to actually come up with the whole genome. Okay, so that's a harder problem. Error correction can be viewed as a front end to the actual problem of sequence assembly. So why is this problem important? And I'm quoting here from 2 bioinformatics domain literature, 2 papers. And so one thing that has been observed is really when you do subsequent steps such as short-read mapping, with genome assembly, what matters quite a bit is the quality of the reads that you put into the assembler. So if you think about sequence assembly, there's probably like 40 different sequence assembly softwares that you can use and that biologists routinely use. But what really ends up mattering is what is the quality of the reads that you use for the assembly. Okay, so that's a very important factor, and so this problem is very important in that sense, and has attracted quite a bit of interest in the bioinformatics domain. Okay, so what is the challenge here? So I showed you this figure with the actual genome and these reads that are arranged in a very specific way. And the problem is it would be nice if this is what we saw. Unfortunately, we don't actually see a picture like that. What we see is all these reads just stack one on top of the other, and we've lost any alignment, or we didn't have any alignment information to speak of in the beginning. Okay. So the question is, you're given these set of reads and you need to correct them somehow, and of course you don't know the true genome. If you knew the true genome, then this problem doesn't exist. Okay, so now just to emphasize, if you knew the picture that you had before, so or if you could tell the sequencer that go ahead and sequence the genome from this particular position to another position number of times, then it would roughly, you could obtain numerous copies of the read and you could use majority logic to decode. So this would be roughly decoding a repetition code. So, and as you can see here, you don't have that luxury here, right? So there's no alignment information, and so there's basically going to be roughly maybe low or no redundancy of the data. So any kind of error correction algorithm requires redundancy of data. Right? So you need to have observed some data multiple times before you can correct it. So we don't have redundancy of the read data, so what do we have to play with? So what we have to play with is these things called k-mers. So what I did in this slide is out of these 6 reads that I have here, I have just indicated the position. I've indicated how many times I saw a string, which is a string of length 3, which is APE. So we don't have, so out of 6 reads, I was able to observe this substring 3 times. So we do have redundancy at this substring level, or what I call the k-mer. So these are called, these substrings of contiguous substrings are called k-mers in this domain. So the key idea is that we can gain redundancy at the k-mer level, and we should be able to use that somehow to perform the correction. So that's the first idea. The second idea is you notice that these k-mers are overlapping. So if you have a read, it is going to be consisting of many different k-mers. So if you are going to think about correcting k-mers, you don't want to correct them in isolation, because if you correct one k-mer, it is going to end up affecting the k-mers that overlap on that point. Okay, so you want to perform this correction of k-mers in a consistent manner, and you'll see that actually sequence detection is a very natural fit for this problem. And the third idea is to effectively exploit the statistical properties of errors. Okay, something that previous methods weren't doing, and this is done through AQM modeling. Okay. So why is this? Why is posing it in this framework a good idea? Because, like I said, actually, to Srinivas asked that question. So there are certain very clear characteristics of next-gen sequences. There's increasing error rate with read position, and there are different substitution rates. So A gets confused with C at a higher rate than B being confused with D, and some platforms also produce insertion deletion errors. Also, although in this talk I'm only going to talk about substitution. And then there are sequence specific errors that have also been exhibited by many of these platforms. So here's before I get into the actual method, so I'll tell you the set of methods that we've compared against. So there's 5 different methods in the bioinformatics domain starting from 2009 to 2013 that we've actually compared our results with. And 3 of these methods are combinatorial methods which don't really use any statistical properties of the sequencer, and 2 of them are statistical. And 4 of these methods assume a constant error rate for all positions. Okay. All these methods assume that the errors are independent across every location in the read. They also assume uniform coverage, and coverage here refers to the average number of times a given base is covered by a read. Okay, so for a given base, if you assume that the start point is uniform, which is not quite true, you can say that the coverage is the number of reads times its length divided by the genome length. Okay. And 2 of these methods assume that quality scores are exact, and what that means is that the sequencing machine, like let's say Illumina, the vendor provides a data sheet that says what the quality scores really mean. What we've seen is actually that is not quite correct. So, and that also causes some problems which I think when we try to model it from the data itself, it helps. Okay, so here's what our technique does. You have a read set, so there are n different reads. The x's are the base calls. So let's suppose that the leads are of length 100. Then each xi is going to be of length 100. It's going to be a string from A, C, G, or T. It can also be an n, and n here stands for erasure. But let's assume that right now we're not thinking about erasures. The yi's are the corresponding quality scores, which means that for every read there is also 100 length vector that says what is the reliability that the sequencer associates with a particular base call. So for the first location, if it calls it as an A, there will be a reliability if it is a number between 1 and 40. So 40 means it's very confident in the call, and 1 means it's not confident at all. So our hypothesis is that each read is generated as an independent realization from the same underlying hidden Markov model, and this is fairly well motivated because true genome sequences are known to exhibit Markov dependence, and the Illumina sequencer, which is the one that I'm going to talk about in this talk, is known to exhibit context sensitive errors. So the probability that it makes an error at a particular location depends on what it has sequenced like 1 or 2 bases in advance. So if we are modeling this as a hidden Markov model, then we need to specify the state space, transition distribution, and emission distribution. So for a given length k, the state space consists of all length k strings of up to size 4 to the k. The local dependence is modeled with certain transition probabilities, and so if you are at a state st, so the st is a k-mer, then you transition into st plus 1 if you have this prefix suffix match, and that's probably best illustrated by something like that, something like the figure here. So if you have k equal to 6 and you are a k-mer act gcg, then you're going to be transitioning into something that starts with ct dct. Okay, so in an arbitrary hidden Markov model, you can jump from any state to any other state. But here, if you're a k-mer, you can jump to at most 4 other states, and that you have to respect this suffix prefix match. Okay, I hope that's clear, because if this was the true genome, this would be the true sequence corresponding to it. Okay, and these lines are showing that what is emitted is the last base. Okay, so in this particular case, I don't have any errors. So the G is emitted here, the T is emitted, the C and the A is emitted here. The emission distribution is the third thing in the Markov chain specification, in the hidden Markov model specification. So if I am at a state ST, my emission distribution consists of 2 parts. There's a part that models the base column, and there's a part that models the quality score. So let me just go back here for a second. So if I'm at this particular k-mer, I am going to be emitting my last base. So here I will be emitting my base G, and it might end up as a G, or it could be corrupted as an EC or T. Okay. So the part that models the quality score goes as follows. It's a very simple quality score modeling. So if your true base, if your emitted base matched the true base, then you have a certain quality score distribution. If your emitted base did not match the true base, then you have a different quality score distribution. So you expect that maybe the mean of the histogram of QP0 is going to be higher than the mean of QP1. Okay. The second part of this is it models the base column. It says that if you are at a particular k-mer, you will be emitting something. You will be emitting one of four possibilities, A, C, G, or T, and those probabilities will sum to 1. But in addition, you have this particular indicator function that I have here, and this is I'll explain this in a little bit more detail later. But it allows us to constrain the overall search space that we are looking at. So this is roughly saying that a k-mer a true k-mer can emit a certain base only if it lies in the D neighborhood of the observed k-mer. Okay, and I'll come to that with an example later. So these are all the parameters that we are going to be estimating when we look at the data from the for the hidden Markov model. And so here's an example. So was there a question? Oh, no, sorry. Okay. All right. Okay. So here's an example. So let's say, for instance, you have an observed tree which is ATTGGCA. This is the one that I've shown here. It could be the result of the transition between these four k-mers of length 4. So for instance, here the first k-mer matches exactly what you're observing. In the second one, the true k-mer could have been TTGC, but you emitted the C as a G, so there's an error at this point. And then from this point on, your emissions are actually matching your observation. So you're emitting an A and you are at an A. So this is an example where the true sequence could be different from the observed sequence by just one error. Okay, so what I would like to discuss now is so we've formulated this problem as one of estimating the hidden Markov model, but there are significant non-trivialities with doing it. So if you do it in a naive way, then you can the problem can quickly become intractable, and you don't get the kind of error correction results that you want. So there are some issues that I would like to discuss. The first issue here is that you can't really choose the state space to be the set of all possible length strings of length k. So even if k is like length 15 or length 20, which is very common, your state space just explodes. You can't do it on a computer, for instance. So the solution to that, what we do here, is to only choose k to be the set of all observed k-mers. So clearly there are going to be some true k-mers, there are going to be some error k-mers, and the hope is that the hidden Markov model is able to differentiate between them. The second problem is the choice of k. So now we have to choose the length k before we start defining this model. And if k is too small, you will realize that a given k-mer. So for example, if k is of length 2 or 3, you can have at most like 4 to the 3 possible k-mers, and you're going to have these k-mers exist in multiple locations in the full genome. So that is going to create a lot of possible paths that explain a given rule. Okay, so you want, our overall goal is to choose k such that most k-mers occur in unique locations in the genome. Okay, and so if you have some idea of the genome length, you can choose a k accordingly. So for example, something that is approximately logarithmic in G, or like 100 times logarithmic in 100 times the length of the genome logarithmic in G, is going to give you something that's approximately correct. Okay, so you are going to have so the assumption here is that most k-mers are going to come from unique locations. So on the other hand, you can't have k to be too high, because you also want to be able to estimate your model. So k can be as long as the read length, and if you have k to be as long as the read length, then you don't have enough data to model your statistics. Okay. The next constraint that I talked about is something in the emission distribution. So we only allow k-mers within the halving distance d of k to have non-zero emission distributions. So I'll explain this by means of this figure. So again, so you have a certain set of emissions here, and the question is, what are the true state sequences that could have explained this emission? So here, the first chamber is ATPG. So you could, if D is equal to 1, then you can have these two chambers lie within the one neighborhood of this guy, so they can explain it. If you are at ADCG, you could transition to PCGG, which is allowed because it allows this suffix prefix match. And this guy here differs from the observed chamber only one location. However, when you another possible valid state transition could have been to PCGA, but we will not allow this. The emission distribution will zero this out because this actually differs from the observed chamber in two locations. Okay, so why is this done? This is done for complexity reasons. So this is otherwise a valid state sequence, but it is not going to be considered in the decoding. So roughly speaking, we can assume this parameter D to be enforcing some form of bounded distance decoding. So we're not going to be looking too far away from the observed state sequence. Okay. Now the last key point here is one of parameter fitting. So if you go back to your Rabiner tutorial on expectation maximization for fitting HMM parameters, we know the Baum-Rich algorithm, that's very well established. But if you do that directly here without enforcing this belief that chambers come from unique locations in the genome, you have this overfitting problem in this situation. So what we actually do is that the EM algorithm maximizes the first term here is the likelihood of observing the read given a certain set of parameters. The second term here is minus lambda, which is a positive value, and j theta, which enforces an L naught like penalty on these transition distributions. So I've just done a MATLAB plot of one of these terms here. So this is a plot of this particular function for different values of p and different values of gamma. So for instance, if gamma is, for example, 10 to the power of negative 18, this is a little problematic. It was 10 to the negative 18. It is almost looking like an L0 objective function in addition to having the properties of different frequencies. Okay. So what this means is that if you are going to make a transition probability non-zero, you are going to be taking a hit of negative lambda in this objective function. So you better have very good evidence that it contributes to the likelihood of the reads. Okay. So following this process, it's the standard thing. So once you've fitted the HMM parameters, you will find the maximum likelihood path corresponding to a sequence of Xi's and Yi's, and you declare the ML path to be the new corrected read. Something that we found was, instead of doing it just once, if you do it multiple times, that helps. Multiple iterations, something like the turbo scheme. This also provides a fairly non-trivial gain in performance. So here are some results. So we have two data sets. One is the C. elegans chromosome 1 and the other is the E. coli. The first one, the genome that we are sequencing is of length about 15 mega bases. The B here is bases. And the first one is, the second one, E. coli, is 500,000 bases. Okay, and the read lengths here are 100 base pairs each. And error correction, I guess I don't really need this slide here, but essentially we are going to be comparing them with what is the effective number of errors that are removed from the set. So here's a comparison with the 5 different methods that I talked about earlier. So eta is the overall gain. So our method is able to correct 98.38% of errors that exist in the database, whereas the closest competitor is the reptile here, which is about slightly over 2% smaller. So these 2% numbers of course need to be scaled by the total number of nucleotides that we are looking at. So it actually turns out to be a fairly significant gain in terms of the number of nucleotides that we are correcting. The second, the C. elegans database, this is an organism that actually has some repeats, and we can talk about that a little bit more later. But here we have a gain that is approximately 3% over the closest method. Some observations. So it's basically a fully probabilistic, and it's a reference free model for next-gen sequencing error correction. We don't assume any constant or independent errors, and so we actually model this context sensitivity that gives us the gain. And we basically don't have any training data here, and we are able to. This HMM framework allows us to effectively deal with this lack of alignment that exists in this DNA sequencing problem. So I just talked about a fairly straightforward emission distribution, but if you invest more energy in thinking about more sophisticated emission distributions, the gain does go up. It can result in an additional 3 to 4% gain depending on the data set. And more recently, we've been looking at insertion and deletion errors also, and that requires a little bit of a change in mindset. We need to augment the state space with what I call K+1 merge as well. But our current results in that domain are also very promising, so we can actually beat the current state of the art algorithms in the bioinformatics domain. So that kind of concludes my first part of the talk. So are there any questions or anything about this particular part, or should I move on to the next part?
I think that's good.
I think okay. So okay, so the second part is, I guess, a completely different application, but so this is about atomic force microscopy. But the essential principles are still the same. So what is this idea of dynamic mode AFM? So what happens is, say, this cantilever is so AFM is used in a variety of domains. So it can be used for imaging, including very soft matter like biological cells and soft polymers, etc. So actually, let me go back to the schematic one more time. So you see here that the cantilever is shown to be in constant contact with the medium. So you can do this in certain situations in which these samples are robust enough to be touched on or scraped upon. But if you have very soft matter like biological cells or soft polymers, you cannot actually run your cantilever over the surface. So what you need to do is to do what is called tapping mode AFM or dynamic mode AFM. So in this case, what you do is you force this cantilever sinusoidally at a certain rate, and this is going to gently tap the medium. Okay, so when your cantilever starts to come in contact with the sample that it is trying to image, it's going to result in some changes in the cantilever depicted. So maybe the amplitude would change, the phase would change, or the frequency of the oscillation would change. And the imaging is done by trying to detect these changes in the parameters of the sinusoid. If you spend enough time and energy, you can actually think about this entire AFM imaging problem in terms of systems viewpoint. So just to explain what these different things are in this block diagram, so if you just look at the upper part of the figure, which is in red, this parameter GT is the parameter that actually is the signal that's used to force the cantilever. Okay, this G omega models the cantilever transfer function. Okay, and PT here is the position of the cantilever, and nu T is modeling some kind of noise. So YT is the noisy cantilever deflection signal. So from a linear time invariant systems point of view, if this stuff in the blue didn't exist, okay, if you put in a sinusoid, you would get out a sinusoid, okay, with the amplitude and phase being modulated by the transfer function. Now, when the cantilever starts to engage with the medium, you have this feedback system coming into play. Okay, so depending on the position and the medium height, the cantilever starts to interact with the medium, and this is usually modeled by a nonlinear block 5. So this looks something like the stuff that I have on the right hand side. This comes from what are called van der Waals forces. There's a repulsive regime and then an attractive regime, and this starts to affect, so this is in feedback, so it starts to affect the position of the cantilever. So what happened at a particular time, its effect is going to affect the later cantilever positions as well. Also want to say that typically this force phi is unknown, this interaction force. Okay, and so you need to decode still in the process in the presence of this nonlinearity. So the whole game in AFM is that you are observing this YT, which is the noisy position of the cantilever, and you want to infer what the medium height DT is. Okay. Now this is conventionally viewed as a slow method, and the reason for that is as follows. Typically these cantilevers that are used are what are called high quality cantilevers, so which means that the time domain impulse response of this particular system is very long. Okay, so what this means is that if the cantilever starts to engage with the medium at a certain point, okay, the effect of that interaction goes through this long time domain impulse response and stays on the medium for a very long time. Okay, so conventionally, what the idea was that if you're using dynamic mode, if you're imaging, you want to move your cantilever laterally in a very slow way. So that you first interact with the medium at one point, and then you interact with the medium again, but you give it enough time so that the effect of the previous hit has gone away. So that was the conventional viewpoint. But once you start thinking about it from a systems viewpoint, you can do stuff that's more sophisticated. So for instance, here one could build a linear observer that is matched to the state of the cantilever. Okay, so imagine that suppose that there was no interaction here. One could build a linear observer that is matched to the state space model corresponding to the cantilever that you can make, and you can get the innovation signal. So in the absence of any interaction with the medium, you would expect that this guy is going to be a mean white Gaussian process. Okay, and the overall effect it has is you can say it's going to reduce the length of the overall channel response. Now, so this is an example, of what something like this can do for you. So this curve that I have here is the envelope of a sinusoidal signal. Okay, that is the envelope of the sinusoidal deflection signal. Okay, and this is the innovation signal that you get out of this observer architecture. So conventionally, what people would do is just look at the deflection signal and try to image. And you can see that there is some kind of change here and some kind of change here. But in fact, if you look at the innovation signal, you see that the innovation signal spikes at 4 different points. So that is this one, and this one. So you see that what this means is at this point the observer is able to track the state of the system, and at this point there is some interaction with the medium, so the observer loses track, and then it comes back into track and then goes out of track again. So the bottom line is that if your objective was just to detect that media interactions instead of chasing the exact medium height, then you can use this idea of a cantilever observer architecture to detect rather than know the precise heights. And this is very useful in many biological experiments. So this will certainly reduce the number of cycles for detection. However, even with this method, you need to wait until the cantilever settles down in its trajectory. And so the question is, and so this was actually work done by Muthi Salavata and co-authors, this was a while ago. And the question is, can we do better than this? Can we improve the speed even more? Okay. And so the whole idea again is one of sequence detection instead of detecting. So for example, here we can think of this method as as detecting interaction by interaction, because you can clearly resolve this interaction in the naked eye. But the idea once again is one of using sequence detection, because what you can do is you think about breaking this feedback loop, okay, and you replace this entire channel by a system that has longer memory but finite memory. Okay, and you think about detecting sequences of interactions rather than interaction by interaction. And so here there are several challenges. So there is this nonlinearity in the feedback loop, which is almost impossible to precisely model, and the nonlinear dynamics of the cantilever also introduces some inherent system memory. So part of it you can model as an intersymbol interference channel, but some of it you can't. There's also other differences. So in this cantilever system, if you think about doing experiments, if you have a sequence of hard hits, that actually causes the amplitude of the cantilever itself to go down. So that changes the input of your channel itself. Measuring these interaction forces, the actual force that is experienced by the exact force that is experienced by the cantilever itself is a very difficult task. Okay, so how do we get around this problem? So here is our proposed channel model for the system. So you have a sequence of bits. You can say that either the medium is high or it's low. This goes through some nonlinearity block. Okay, and this block is probabilistic in interest. You can think of that as a Markov model. This produces a sequence of impulsive forces. Okay, so we don't actually get to measure these, but these produce a sequence of impulsive forces that are then exciting this LTI system. This gamma t is the response to a single impulsive force. It is then corrupted by noise. So what we observe is actually this particular signal. and what we want to infer is the split sequence. Okay, so because you have this additional nonlinearity block here, the traditional detector design strategies actually do not work for this problem. So this is a block that we can actually measure in the lab. So this is impulse response that can actually be measured, and we can actually do that in the lab by measuring the response to one particular impulsive bit. But this part we actually cannot measure, this nonlinearity block. So if you assume that each feature is high or low with probability 1/2, you can actually, I forgot to mention this, that even though what we measure here, we can actually when you're trying to think about this in terms of detection, we can show that a white and match filter is the correct thing to do here, which I already have in this block diagram. So you can say that what we are observing are the ZKs, which are the sufficient statistics for the detection, and what we want to do is to find that bit sequence that maximizes the probability of the conditional probability of observing the sufficient statistics given the bit sequence. So here, since I assume that the features are high or low with equal probability, whether I look at the joint or the conditional, it doesn't matter. And so we need to be able to do this in a tractable manner. So here is the dependency graph of this system. So what you observe are these DIs, which are the sufficient statistics for the detection. What you're trying to detect are these AIs, which are the bits, and the things in the middle are these interaction forces, which are not something that we observe. Okay, so we only observe this guy, and we want to input this guy. And again, I'm skipping a lot of things here, but using some ideas from using the baseball algorithm on this dependency graph and making some assumptions, you can cut down the dependence of this, the ith conditional statistic in terms of finite dependence. In terms of it depends only on the last number of finite number of bits, which is m plus mi, and the last mi previous sufficient statistics. And if you make further assumptions on Gaussianity, which is to say that this vector of sufficient statistics given the state transition, you assume that this is Gaussian, then we can actually reformulate this entire problem in terms of an instance of the Viterbi algorithm with Markov Modeling. Okay, so this is the Kopchik-Morrow algorithm, and the nice thing here is that we can actually measure. So if you want to compute, let's say, the branch metrics of each of these transitions, these are things that actually we can measure in the lab. The sufficient statistics are something that can be measured, and you can get the mean and the covariances of every branch metric by having access to training data. Okay, and here in this case we do need training data. So of course, for a general image, the features are not going to be equally likely to be high or low, and so in this case we can actually reformulate the problem instead of just ML sequence detection as a map symbol detector. So it's essentially like the BCGR version of the ML sequence detection problem. So here instead what we are doing is, given all the sufficient statistics, we try to detect whether a given bit is high or low. And how do we change the prior? So I can explain that in just a moment. I'll come to the adaptation of the prior shortly. So here's an experimental setup. So, and these are all results that were obtained in the lab. So these are the parameters of the cantilever. This is a test pattern that was made in the lab. This is 2.7 micrometer by 10 micrometer. So 10 micrometers in this dimension and 2.7 micrometers in this dimension. And I'm showing here results from 4 different techniques. These are the 2 B and C are what you would find in a commercial AFM, for instance. This is the method that corresponds to the cantilever observer architecture, and this is the method which we call BCGR imaging. And as you can see, this particular method provides a significantly better image than any of the other techniques. Okay, now what I mean by adapting the prior is that when you're scanning this particular reference sample, in the beginning, for the first line, you can assume that the priors are equally likely to be, they're equally likely to be 0 and 1. But once you decode the first line, you can assume that to be you can assume that your decisions are correct, and you use that as the prior for the next line, and then you keep moving forward in that way. Okay? And that's how this particular image is obtained. Just to give you an idea, if you wanted this quality of an image, using a conventional technique, let's say such as the amplitude imaging, you would need a scan rate of at most about a lateral scan rate of at most about maybe 3 micrometers per second. So we are getting a speed up that is at least of an order of magnitude in this technique. Okay, and this is like maybe just a blow up of close up of some of the features that we are able to extract. So this is the true, the high frequency content in this image. This is the true reference pattern. This is kind of gone in the first two methods. It's recovered somewhat in the cantilever observer architecture, but we can actually reconstruct it very well at this particular scan rate. So yeah, so just to kind of, so in the second part of the talk, I mean, there's a lot more details that I could have probably gotten into, but I chose not to, and I can certainly talk about that in more detail if you want. So just to, I mean, some observations here that the systems viewpoint is actually very useful in dynamic mode AFM based nano-imaging as well. So, and I guess the main difference here is that this mindset of detecting interaction rather than chasing the exact height is actually quite useful, and it's not only useful for speed reasons. There are application domains where detecting rather than finding the exact height is very useful. For example, like video rate imaging of biological phenomena. And we have some extensions to multi-bit imaging that I did not talk about here, but as you can expect, the problem does become progressively harder. And there's actually a very nice connection between a lot of techniques in signal processing and what I call here bioinformatics. So basically, there are some very nice connections between blind equalization, some classical stuff in blind equalization, to the problem of not only imaging but also trying to estimate material properties, and that's something that is very recent that we've been doing. Hopefully, I'll talk about at some other point. So that more or less brings me to the end of this talk. So these are the collaborators. Dushyanian is a student in bioinformatics and computational biology, and also in statistics. Zhao Som is a former student of mine who's now at DU. And Dr. Dorman is in statistics and also genetics development in cell biology. So he's a faculty member in that department, and the collaborators. We were collaborators on the DNA sequencing work. Naveen Kumar was a former student of mine who now works in the Bay Area, and Pranav Agarwal was a student at the University of Minnesota, and Murthy Salapaka is a faculty member at the University of Minnesota. So they were the collaborators on the nano-imaging work. So yep, so that brings me to the end of my talk. I was budgeting for about 45 minutes. I think probably about right. So okay, so that's it. Thank you.